So another major heading under which we're going to study why these RBCs have abnormal shape and size is damage due to the RBCs after the formation. The reasons behind this can be either intrinsic or extrinsic. In intrinsic factors, we are going to study about different hemoglobinopathies and membrane defects due to the enzymes, because that leads to more fragile boundaries of the RBC, because of which they lose their shape. And extrinsic factors in which there is influence of drugs, chemicals, toxins, heat, and abnormal forces. So under this heading, we are going to study about hyperchromasia, spherocytosis, and irregularly contracted RBCs and elliptocytes or ovulocytes. Further elaborating about hypochromia or hyperchromasia, in this we usually see more deep staining red cells with a lack of central pallor. It is seen in either in cases of macrocytosis caused in the cases of neonatal blood, megaloblastic anemia, in which the MCHC is normal, or in case of red cell abnormality, in which the cells are abnormally rounded in case of spherocytes, and in case of abnormally contracted cells in which the MCHC is increased. So, in spherocytosis, what happens is that the cell becomes more like a sphere and loses its disc-like orientation. The diameter is less and the thickness is a little more than the normal. But we need to realize one fact that even though the shape has changed, but their boundaries are still regular. And the irregular boundaries, they represent more of crenated RBCs and are seen in other abnormalities, that is not spherocytosis. It is seen in genetic defects of cell membrane like in hereditary spherocytosis, where there is interaction between the immunoglobulin or complement coated red blood cells and the phagocyte, like in delayed transfusion reactions, ABO hemolytic disease of the newborn, and autoimmune hemolytic anemias, and in bacterial toxins like the ones caused by Clostridium perfringens and snake venom. So the word spherocytosis indicates more like a sphere and less like the elliptical shape of an RBC. And some of them are more blue in color, indicating a high reticulocyte count. And further proceeding on to the apiohemolytic disease of the newborn, spherocytosis is more intense. So compare the shape, and polychromatophilic RBCs are more prominent. The color of the RBCs is more blue, indicating that the reticulocyte count will be more high. So going on to irregularly contracted red blood cells, in this we can see that some of the cells are smaller and are usually more densely stained. See, this is a more densely stained cell and the margins are irregular. This is a normal stained RBC. The margins are regular and this is relatively smaller. Similarly, as you can see in this cell, the margin is irregular. In this cell also the color is more deep and the margins are irregular. So they appear contracted and they are partly concave. In this picture, we can also see one semi-ghost cell. In this, we can see that the chromatin, the hemoglobin, it is contracted towards one side. The other part of the RBC, it is empty. So the hemoglobin is pushed to one end and the rest of the cell is empty. So what are the causes of having these irregularly contracted cells? It can be either due to oxidant drugs or chemical induced hemolytic anemia, unstable hemoglobinopathies like hemoglobin con and St. Mary's hemoglobin, hemoglobin E homozygosity, and rarely hemoglobin E heterozygosity, and copper induced Wilson disease, in which, due to liver toxicity, the RBCs get destroyed by the liver. So, in this image, this is a case of G6PD deficiency. This is the ones being pointed by the black arrows. These are some ghost cells, and we can also appreciate some irregularly contracted RBCs. So see, this is a more deeply stained RBC as compared to this, and the margin is again irregular. It is smaller, it appears smaller, and this is again another irregularly contracted RBC. And we can also see Heinz bodies being protruded out of some RBCs. Like this RBC has an Heinz body over here. Another one can be seen in this RBC. So for it is seen in cases of hereditary eleptocytosis, and a large number of avilocytes or eleptocytes can be seen. So see, these are avilocytes and these are eleptocytes. So they don't have that biconcave structure, the disc-shaped structure of a proper RBC anymore. Then there is another entity called hereditary pyropoikilocytosis, in which a lot of other cells are also seen, and ovulocytes and eleptocytes are a part of the population. See, other cells are also seen, but see, you can see some eleptocytes. See, these are irregularly contracted cells also that can be seen over here, right? And then we can also appreciate some eleptocytes like this, ovulocytes like this one. So it is one of the many cells that are seen in this situation. So further talking about spiculated cells and red cell fragmentation, they are basically in this heading we'll be studying about the four main types, that is cystinocytes, keratocytes, acanthocytes, and echinocytes. So starting with schistocytes, it is also called fragment. It means fragmentation actually. So the cells are usually smaller than the normal RBCs. These are fragmented RBCs. They can either stain more palely or deeply. So see, this is a more deeply stained cystocyte. This is a more deeply stained. This is a palely stained. And see, they have spines. They have spurs, sharp angles or spines, right? So it is seen in case of megaloblastic anemia or dyserythropoietic anemia. In case of burns, microspherocytes are seen. So microspherocytes are both round and dense. So this is an image from a patient of burn, and this is a very deeply stained microspherocyte. The size is smaller than a normal RBC. It is more dense and the edges are still round. So in what all conditions do we see these microspherocytes? It is seen in genetic disorders like thalassemia, congenital dyserythropoietic anemia, hereditary pyropoikilocytosis, in acquired disorders of RBC formation like megaloblastosis and dyserythropoiesis, in cases of mechanical stress like microangiopathic hemolytic anemia, and patients who are on prosthetic valves, and it is also seen in cases of direct thermal injury. Further talking about keratocytes or horn cells, so as it's very evident from the name, they have horns, spicules that are either one or two. They are also called bite cells. And why are these formed? These are formed because of exclusion of Heinz bodies or by mechanical damage. Further talking about acanthocytes or spines, so they have numerous spicules that are of variable length and thickness distributed irregularly throughout the surface of the cell. So they are of variable size, variable length, variable distribution, and it's present in multiple cells, not on one or two cells, because that can also be an artifact. But since it's present on most of the cells, they are called acanthocytes. It's seen in case of abnormal phospholipid metabolism, in inherited abnormalities of red cell membrane protein like McLeod's phenotype, post-splenectomy or hyposplenism, and in severe liver diseases. Further talking about echinocytosis or a sea urchin-like or a hedgehog-like cell, it is also known as crenation, and the RBC develops short regular projections. So in acanthocytes, there were variable number, variable sizes, and irregular distribution. But in echinocytes, they are regularly distributed, short spicules that protrude from the surface of the RBC. It is also seen as an artifact seen in cases of patients where the samples have been stored above 20 degrees Celsius overnight. So, in that case, it's an artifactual finding. It is also seen in conditions like uremia or in preterm infants where exchange transfusion has happened. So, this was all about this presentation. In the next one, we'll be talking about the other erythrocytic abnormalities like leptocytes, Target cells, stomatocytes, sickle cell, hemoglobin C crystals, and SC-poikilocytes, erythrocyte inclusions, Hobble-Jolly bodies, Pappenheimer bodies, Roulet, and autoagglutination. Changes associated with compensatory increase of erythropoiesis, like seen in polychromasia and erythroblastemia, and effects of splenectomy or hyposplenism.